Good morning, everyone. It's just 1 minute past 8, and we are starting the next set of grand rounds this fall in nephrology. So today we are excited to have Adrianna Douvris. Many of you may know her, who was a nephrology fellow with us. She did her MD from the University of Toronto and then decided, smartened up and came to Ottawa for her Internal Medicine and Nephrology fellowship. After finishing her fellowship a few years ago, she has been around doing a PhD at Dr. Bond's lab in Cellular and Molecular Medicine on the stuff that she's going to present today. Presently, she has received a KRESCENT award to pursue a post-doctoral fellowship working in Dr. Stanford's lab on kidney organoids. So welcome, Adrianna.
Thank you. Thanks for inviting me to come and talk about my work. So I'm going to be talking about what I've spent the last probably 3.5 years doing, which is my PhD project titled mir-486 for acute kidney injury and vascular outcomes. And although I spent a lot of time doing research and less time doing clinical work, I just wanted to show here that I did have, one second. I'm hearing an echo. I did want to show here, thanks. I did want to show here that I had little patients, and this is just a picture of me in the animal care facility taking some blood pressure measurements by tail cuff on our rat subjects. That was much harder than taking blood pressure on patients. The rats are very difficult to train. So for the purposes of today, I'm going to give an overview of microRNAs and acute kidney injury to give some background on the topic. And then I'm going to focus in on our microRNA of interest, which is mir-486-5p in our rat model of acute kidney injury. And we'll talk about some of the newer stuff that we've done with long-term kidney and vascular outcomes in rats after AKI. And then I'll end with the last part of my PhD, which is looking at the effect of mir-486 and cultured endothelial cells. So it's not a traditional clinical talk for grand rounds, but I'm going to try to add some clinical relevance wherever I can in the talk. So as an introduction, going back to molecular biology 101, so only about 2% of our DNA in the human genome is actually transcribed into messenger RNA, which is then translated into protein. The other 98% of our genome is classified as non-coding RNA, and this was traditionally thought to be junk RNA, but is now known to have very important function. MicroRNAs are within that category of non-coding RNA. They're short non-coding RNAs, and that's the one that we're going to focus on for the rest of the talk. MicroRNAs were first discovered in 1993 in the C. elegans worm, but then since then, over 2,000 microRNAs have been discovered in humans. MicroRNAs are short non-coding RNAs, about 22 nucleotides in length, that are evolutionarily conserved and important regulators of gene expression. It's actually estimated that over 60% of protein coding genes are microRNA targets and therefore regulated by these mirs.
Very much integrated.
I won't. So microRNA biogenesis involves transcription by RNA polymerase II, which leads to the primary microRNA transcript, and these are further processed to generate pre-microRNAs, which are exported from the cell nucleus, and then the pre-microRNAs are then processed by an enzyme called dicer, and the mature microRNA is loaded onto an RNA-induced silencing complex. So here's a little schematic of how it works. So this is a cell's nucleus, and this is your DNA being transcribed into messenger RNA, and once that's transcribed, it gets exported into the cytoplasm. In the cytoplasm, the microRNA is bound to its RNA-induced silencing complex and binds its specific sequence on the messenger RNA, thereby either inducing the messenger RNA for degradation or inhibiting its translation. And the end result is that it induces post-transcriptional gene repression. One of the other interesting properties of microRNAs, in addition to their function, is their stability in the circulation and other body fluids, like, for example, urine. They've even been measured in donor kidney perfusion fluid from deceased kidney donors. And the reason for their stability is that they circulate either within extracellular vesicles or complex with RNA binding proteins, such that they're resistant to endonucleases. Then they also exhibit tissue-specific expression. And as a result, they're being studied for biomarkers for a wide spectrum spectrum of diseases, including but not limited to coronary artery disease, chronic kidney diseases, acute kidney injury, neurodegenerative diseases, liver diseases, and all sorts of cancers. And so now we're going to shift gears and talk a little bit specifically more about microRNAs in human acute kidney injury. A recent systematic review of microRNA therapeutics in animal models of AKI that was done a few years ago identified 70 studies that totaled 42 microRNA species that were tested mostly in mice. But to get an overview of which microRNAs are studied in human acute kidney injury and how, I conducted a systematic review on the topic. We identified 2,430 studies and included 92 for qualitative synthesis. Here, there were no interventional trials. Every study that we found was a biomarker study or mostly an animal model study that contained some limited clinical data. So just like in the animal world, the microRNAs in human diseases have been a growing field. So this graph just shows the number of publications of microRNAs in human acute kidney injury from 2010 all the way to 2023. There was a little bit of a blip in the COVID years as less research was being conducted on this stuff. But overall, the trend is that it's rising. We also identified several distinct AKI categories in human acute kidney injury, including AKI associated with cardiac surgery, sepsis, nephrotoxins, kidney transplantation, and other causes, which include studies that either had a mixed AKI population where you couldn't discern a single cause, or single studies of specific AKI causes that were less commonly studied, like acute obstruction or acute GN. So all in all, we identified 128 dysregulated microRNAs in human acute kidney injury. And of those 128, only 30 were reported in 3 or more studies. We basically included studies that had targeted approaches where they either measured the included studies had either employed targeted approaches where they measured one or a very select few of microRNAs with qPCR. And then other studies that we found were high throughput screens. From these high throughput screen studies, we only included the microRNAs that they validated from their initial screens, which totaled the 128 that we came up with. The figure here highlights the microRNAs that were identified in 3 or more studies. And one of the interesting ones is microRNA 21. This was the most commonly identified dysregulated microRNA across categories in human studies. And interestingly, timing of AKI onset is frequently challenging to pinpoint clinically, but in cardiac surgery specifically, kidney function can be systematically measured at specific time points, like preoperatively at baseline, and then at specific time points postoperatively. So what we found in our systematic review is that microRNA levels are higher in patients with AKI for most etiologies, but for cardiac surgery, patients with cardiac surgery associated AKI tended to have lower levels of both serum and urinary mir-21. And that actually seems to mirror the animal model data on ischemic acute kidney injury, where mir-21 is also the most frequently studied microRNA. In animal models, generally gene deletion experiments where endogenous mir-21 is knocked down in the kidney demonstrated exacerbation of kidney injury, and animal model studies of ischemic AKI also demonstrated that ischemia-induced upregulation of mir-21 was renoprotective, which might coincide with its mechanism in cardiac surgery-associated AKI. So out of all the mirs that we identified in our systematic review, mir-21 seems to be the most promising as a biomarker and for therapeutic intervention. But we did note in our study that we do need to be cautious interpreting any of these data because there were several biases in our studies that we identified. For example, many of the mir-21 studies in human AKI were targeted approaches because of the large amount of preclinical data on that specific mir. And I wanted to highlight here that 84 out of the 128 microRNAs were only reported in single studies, which suggests right now that reproducibility could be a concern in this field. I wanted to show here that although we didn't find any interventional studies of microRNAs in acute kidney injury, I've actually found 2 in chronic kidney disease. The first one was just recently published in CJASN, and it's a phase 2 trial of anti-mir-21 for Alport syndrome, where participants were followed for 48 weeks. Unfortunately, the study didn't find any difference in GFR decline, but also did note that there were no reported adverse safety effects. So that in itself could be promising. And this second study hasn't been published. I found it on clinicaltrials.gov, but it's a study of an anti-mir-17 compound for polycystic kidney disease. So microRNA therapeutics are coming. To shift gears a little bit, to segue into our specific project, I wanted to touch on long-term outcomes after acute kidney injury. So we all know that AKI is a common complication of hospitalization with a high in-hospital mortality rate. But patients with recovered acute kidney injury are also at risk of long-term complications. These include newer progressive chronic kidney disease, kidney failure, and even an increased mortality risk. And despite the health consequences of AKI and how prevalent it overall is, we know that no effective treatments exist and our preventative strategies are limited. The pathophysiology of AKI also links to the clinical long-term outcomes that we've seen in the literature. So here I'm going to focus specifically on ischemic AKI, but essentially ischemic AKI involves injury to the tubular cells and to the endothelial cells, and pathophysiologically can be described by 4 stages: the initiation and extension phases of acute kidney injury, followed by a maintenance phase where the surviving kidney epithelial cells dedifferentiate and proliferate to restore tubular epithelial cell populations. And then lastly, recovery, where cell and organ function is restored. However, in the course of acute kidney injury, some tubular cells undergo what we call maladaptive repair, which is characterized by profibrotic signaling, and this gives rise to interstitial fibrosis and the progression to chronic kidney disease. Endothelial injury is an important hallmark of the pathophysiology of AKI because it results in several bad things happening. So it increases leukocyte adhesion, shown in this cartoon over here. It increases vascular permeability, it causes abnormal coagulation, and it's pro-inflammatory. And all of the above mechanisms contribute to decreased renal blood flow. And these effects can serve to extend tubular injury. But not only that, they also lead to a permanent reduction of kidney vascular density, resulting in intrarenal hypoxia. So consequently, endothelial injury is another important contributor to interstitial fibrosis and the transition to chronic kidney disease. In order to study AKI in animal models, there are several experimental models, but all of them really study the renal causes of AKI, including ischemic ATN, nephrotoxic ATN, and sepsis. The model that we are focusing on in our lab is the ischemia reperfusion injury model, which is induced by bilateral renal pedicle clamping, shown here in this cartoon. And this model is relevant to very specific clinical scenarios that include cardiac surgery and delayed graft function and kidney transplantation. So before going on further with AKI, I'm going to switch gears and introduce mir-486-5p and what it is and kind of where it came from. So mir-486-5p is our microRNA of interest. Here, it's just to show some of the background on it. It's actually transcribed from an intron within the ankyrin 1 locus and then processed to its mature form, which I won't get into details about. But it was actually first discovered as being downregulated, so differentially expressed in Duchenne muscular dystrophy. And because of the promoter that drives its expression, it's actually highly expressed in skeletal muscle, cardiac muscle, and in red blood cells. The expression in the kidney itself is actually not very high. Given its high abundance in heart muscle, it is not surprising that it's been studied in cardiac ischemic models. So a few studies have come out on it. For example, in animal models of cardiac ischemia reperfusion injury in mice, mir-486-5p was found to prevent cardiac injury. And then in another experimental model of myocardial infarction by coronary artery ligation in both mice and non-human primates, mir-486 was shown to preserve heart function and to preserve cardiac blood vessel density. The mechanisms aren't completely known, but they do implicate mir targets that are involved in the regulation of apoptosis, which is otherwise known as programmed cell death, and angiogenesis, which refers to the growth of new blood vessels. So where did we come up with mir-486? So mir-486 came from earlier ischemic AKI experiments in Dr. Burns's lab that were done in mice that utilized cell-based therapies and extracellular vesicles as preventative treatments. It was found that mir-486 was highly enriched within protective extracellular vesicles and was therefore hypothesized to be the mediator of protection against AKI. In an earlier study, when we administered mir-486 to mice with kidney ischemia reperfusion injury by tail vein injection, we found that the injection of direct mir-486 reduced ischemic AKI and also inhibited the expression of genes that were involved in inflammation and apoptosis, specifically within proximal tubular cells. But we still didn't have any knowledge of what the long-term implications of this therapy are. And we also suspected that it has some protective effect on endothelial injury as well, but we were not sure what that was yet. So that kind of led to the objectives of my PhD project, which was to study the effect of mir-486 on the renal endothelium, vascular function, and long-term outcomes. The rat model was more ideal to answer these questions because it's a better model to study vascular function. So we specifically were looking at the effect of mir-486 on endothelial injury in the short term, but also the long-term kidney and vascular outcomes after treatment. And then we have another part of the project where we looked at the effect of dosing and timing of administration. And so how did we do this? To determine the effect of mir-486 on rats with ischemic AKI, the rats were subjected to bilateral renal pedicle clamping for 45 minutes, followed by reperfusion, which is when we unclamp. Liposomal packaged mir-486 or a placebo scramble microRNA was administered by tail vein injection at the very start of reperfusion, and then outcomes were assessed after either 24 hours or 10 weeks for kidney function, histological analyses, and vascular function. So here we performed in situ hybridization to localize mir-486 within the kidney cortex 24 hours after kidney IR injury. Mir-486 signal, hopefully it shows up okay here, looks like small little pink-red spots. These little spots can be seen within proximal tubular cells. And interestingly, we see signal both in the cytoplasm, which is like the whitish area, but then also in the nuclei, which are stained blue. Mir-486 signal was also detected within interstitial endothelial cells, which you have to really increase the magnification to see, but it's in this bottom image. We then evaluated regional renal blood flow by laser Doppler flowmetry 24 hours after kidney IR injury and treatment with mir-486. To do this, we anesthetized the rats with an inhaled anesthetic called azoflurane. We made a little left flank excision to expose the left kidney, which we had to decapsulate. And then using this laser Doppler probe, we first sat the probe on the kidney surface to measure cortical blood flow, and were then able to insert the probe into the kidney parenchyma to measure the medullary blood flow. What we found was that cortical blood flow was decreased in kidney ischemia reperfusion injury, but it was preserved by mir-486. And we also found that mir-486 significantly enhanced medullary blood flow compared to the rest of the groups. And we suspect that this should have functional implications for its protection against ischemic AKI. Plasma creatinine and blood urea nitrogen levels were measured at 24 hours and 10 weeks after kidney IR injury. So rats that with kidney ischemia reperfusion injury had a significant rise in both their plasma creatinine and BUN levels. And this was profoundly attenuated by mir-486. The creatinine was basically exactly the same as the sham operated rats. At 10 weeks, kidney function had normalized and was similar between all groups. So we found no differences in kidney function long-term. We also looked at histological injury by periodic acid shift staining of rat kidney sections. Here, we show that kidneys from rats subjected to ischemia reperfusion injury had obvious evidence of acute tubular injury. The arrows and asterisks are depicting tubular casts, tubular dilatation, and nuclear loss, which are all signs of acute tubular injury. We noted significantly less tubular injury in the kidneys from rats that had been treated with mir-486. So When endothelium becomes injured, it also becomes activated, which is a process whereby the endothelial cells express adhesion molecules that facilitate the attachment and migration of leukocytes into the injured tissues. We looked at one of these adhesion molecules known as intercellular adhesion molecule 1, or ICAM1, in the rat kidneys at 24 hours after kidney ischemia at reperfusion injury. And the immunofluorescence here shows that ICAM1 should stain red, and the green stain is staining the blood vessels or peritubular capillaries. We show here that ICAM1 signal is upregulated after kidney ischemia reperfusion injury, but not in the rats that were treated with mir-486. To confirm this by a different method, we also performed immunoblotting for ICAM1 and whole kidney lysate, which also shows this ischemia reperfusion-induced upregulation of ICAM1 that is inhibited by mir-486-5p. And then since mir-486 inhibits endothelial activation induced by ischemia reperfusion, we looked at kidney inflammation by immunohistochemistry as well at 24 hours. We stained, so here we're staining for neutrophils, and the neutrophils stain brown in this picture. We show that there's increased neutrophil infiltration in the kidneys of rats subjected to kidney ischemia reperfusion injury at 24 hours, and that neutrophil infiltration was significantly decreased by mir-486. So in addition to studying the effects of mir-486 on AKI and vascular function in those first 24 hours, we also conducted analyses at 10 weeks to look at kidney outcomes and systemic vascular outcomes. So after 10 weeks, we found that the rats that were subjected to kidney ischemia reperfusion injury had increased picrocereus red staining, which stains for collagen. And this indicates increased collagen deposition, which is a marker of tubulointerstitial fibrosis. Again, the rats that had been treated with mir-486 at the start of reperfusion had significantly less collagen staining, suggesting that treatment with mir-486 prevented long-term increased collagen deposition and fibrosis. Next, after those 10 weeks, the rats that had been subjected to kidney ischemia reperfusion injury had about a 30% reduction in peritubular capillary density. And this is indicated by the brown staining of peritubular capillaries. Here, we show that peritubular capillary density was largely preserved in the rats that had been treated with mir-486. Next, we looked at systemic endothelial function, importantly because we know that endothelial function and impaired vasorelaxation is a predictor of cardiovascular risk, and this is something we can directly examine using mesenteric vessels. The mesenteric bed is shown in this image over here. So this is a bit of a difficult protocol. I'm pretty happy that we got to be able to do it. The mesenteric arteries have to be dissected very carefully from the mesenteric bed so as not to injure the endothelial layer. And then using a microscope, the vessels are carefully mounted onto a wire myograph that's connected to a force transducer. And so this is able to record vessel constriction or relaxation in response to drugs to determine the degree of endothelium-dependent vasorelaxation. I didn't show this data here, but we did measure blood pressure in our rats, and we found no difference at 10 weeks, but we did see a difference in vessel function. So from the curves, a healthy endothelium or a vessel with healthy endothelium will relax normally, and the curve should shift to the left and downwards. Abnormal endothelium should follow a curve that shifts upwards and to the right. So from this graph, the blue line represents mesenteric arteries from sham-operated control rats. And then we see that the mesenteric arteries from the rats that had been treated with mir-486 dilate very similarly to the sham-operated group. On the other hand, the mesenteric arteries from the other post-ischemic rats, those curves separate, and they exhibit impaired endothelium-dependent vasorelaxation. And those are the red and magenta curves. So in summary, mir-486 administered at the start of reperfusion to rats protected against ischemic AKI, preserved regional renal blood flow, protected against endothelial injury and inflammation, and long-term prevented against the transition to CKD and protected against systemic endothelial dysfunction. However, we did find an unexpected finding while we were conducting this research, and that involves a very important enzyme called endothelial nitric oxide synthase. This is a key enzyme in the production of nitric oxide, which is a vasodilator and a very important regulator of kidney microvascular blood flow. We had expected, we measured endothelial nitric oxide synthase because of the protective effects that we were seeing on endothelial health and on regional renal blood flow. So we looked at eNOS levels by immunofluorescence, and this is an immunofluorescence image of a renal arteriole where the smooth muscle wall stains green and eNOS stains red. Kidney IR injury actually increased eNOS protein levels, and mir-486 inhibited this ischemia-induced upregulation of eNOS. We also verified this result by performing immunoblotting for eNOS protein and whole kidney lysate. Again, this shows that eNOS is upregulated by ischemia and that mir-486 inhibits the ischemia-induced upregulation of eNOS. Next, to determine whether this effect was due to a mir-486's function within endothelial cells, rather than an effect that's simply associated with the prevention of kidney injury, we looked at the effect of a later administration of mir-486. So to begin to address this, we had a very small study in a small subgroup of rats where we subjected them to bilateral renal artery clamping, and 4 days of reperfusion. We injected, or we treated a group of them with mir-486, and then we collected the kidneys at 5 days after kidney ischemia reperfusion injury, on 1 day after mir-486 treatment. Again, what we saw here is that 4 days after kidney injury, eNOS protein levels remain increased compared to the sham-operated controls. However, the later administration of mir-486, 4 days after kidney IR injury, actually still decreased eNOS protein levels. So this suggests that mir-486 decreases eNOS protein levels in endothelial cells, although we didn't see any adverse effects in vivo with a single dose of mir-486 administered at the start of reperfusion. To explore the significance of this in vivo further, we conducted another study with rats where we looked at the effect of both delayed and repeated administration of mir-486. And so to do this, we subjected rats to bilateral renal artery clamping, followed by reperfusion, and we administered the first dose of mir-486 at 96 hours of reperfusion and a second dose at 3 weeks. And then we looked at the same outcomes. What we found was that delayed mir-486 did not protect against the development of tubulointerstitial fibrosis. Collagen deposition was similarly enhanced in all 3 post-ischemic groups of rats, and delayed mir-486 had no impact on preserving vascular density at 10 weeks. What we also found was that mir-486 didn't worsen any of these outcomes. We didn't see more fibrosis or even further reduction in peritubular capillary density. We also performed mesenteric artery wire myography on this group of rats as well. And we found that the delayed mir-486-5p administration had no impact on preventing systemic endothelial dysfunction. All the mesenteric arteries from all groups of post-ischemic rats demonstrated impaired endothelium-dependent vasorelaxation. I also wanted to note that the vessels that were from the mir-486 treated rats didn't have any additional impairment. So in summary, late administration did not improve and did not have any additional adverse effects on kidney or vascular outcomes. Basically was a completely negative study with no effects. In order to in order to further understand the mechanism and impact of mir-486's effect on eNOS expression, we turned to an in vitro model of cultured endothelial cells. Because as we know so far, the mechanism and impact of mir-486 inhibition of eNOS is unknown. So we cultured human umbilical vein endothelial cells, and we treated them with the same mir-486 mimic that we used in our rats and the same scramble or placebo microRNA that we used in rats. And then we analyzed protein, RNA, and endothelial cell functions. So looking at, we looked at eNOS messenger RNA levels by qPCR, and we found that cells that were treated with mir-486 had significantly decreased eNOS messenger RNA levels. And then we looked at eNOS protein levels by immunoblot and found the same thing, that mir-486 significantly decreased eNOS protein levels. So this is consistent with what we saw in the rats. Next, to determine the effect of mir-486 and the impact of eNOS downregulation on endothelial cell function, we performed functional assays. And the one that I'm going to present here is the major gel-based network formation assay. And this is used for the in vitro assessment of angiogenesis, which is the process by which new blood vessels are formed. So tissue culture plates are coated with a substance called major gel, which contains, it's a polymeric substance that contains a bunch of growth factors. The endothelial cells are seated in the major gel, and then they're allowed to form networks that are composed of branching nodes. And at specific time points after seeding, they're imaged under the microscope to have a look at what the network looks like and to quantify this. What we find here is that compared to the untreated cells and the other controls, the cells that were treated with mir-486 had less nodes and less branching. We also selectively knocked down eNOS and endothelial cells to look at that phenotype and found the exact same thing that we see with mir-486, this inhibition of network formation. To determine the impact of eNOS downregulation on mir-486's inhibition of angiogenesis, we performed a rescue experiment where we transfected cells with mir-486 and an eNOS plasmid to restore eNOS expression in the cell. Again, mir-486 inhibited network formation, as shown in the image over here, but this inhibitory effect was rescued when eNOS was restored. And so this suggests that mir-486 inhibits angiogenesis in an eNOS-dependent manner in endothelial cells. So mir-486 inhibits angiogenesis through eNOS inhibition, but now what? How does it do that? So there are databases online that predict microRNA targets based on the gene, based on the sequence of the gene's 3' untranslated region and its homology to the microRNA sequence. And there's about 300 predicted targets for mir-486, depending on the database that you choose. eNOS is not a predicted target, but that doesn't mean it can't be a target. So we performed an assay called the luciferase assay to determine whether there was direct targeting of eNOS by mir-486. And this assay is actually based on how fireflies generate light and glow. So basically, this assay measures light production. So light production requires an active luciferase enzyme. And so we transfected cells with a luciferase, with a reporter vector that contains the luciferase gene that's fused to the eNOS 3' UTR. And we transfected this along with mir-486 to the cells. If a mir-486 binds to the 3' UTR, it would inhibit the production of luciferase, which would therefore inhibit light production. And if mir-486 doesn't target the 3' UTR, then light should be emitted normally. But unfortunately, fortunately or unfortunately, what we saw was that mir-486 had no impact on light production. And so we determined that it's not a direct mir-486 target, and that there must be some other mechanism by which mir-486 seems to modulate or regulate the expression of eNOS. So I had to find a way to screen for mir-486 targets in endothelial cells, and found a method that was interesting from the literature, where you can literally pull down your microRNA of interest, bound with hopefully all of its target I found a method in the literature where you can pull down your microRNA of interest along with its bound messenger RNA targets. And the pull down RNA can be purified and it can be analyzed either by microarray or RNA sequencing. And so we decided to do this by RNA sequencing. So we transfected biotinylated mir-486 into the endothelial cells, and then we pulled it down, bound to its messenger RNA targets. We purified the RNA, and then we sent it for RNA sequencing at the STEM core laboratories at OHRI, and then validated some of the data. And what we found were that a lot of high abundance transcripts were identified in the pull down RNA. So this figure is a volcano plot of all the differentially expressed genes in the mir-486 pull down relative to an appropriate experimental control. The differential expression is calculated by a log 2 fold change in the mir-486 pull down compared to our control. In red, I'm highlighting predicted targets from one of those online target prediction databases. And all of the other colors, the greenish to blue colors, are genes or transcripts that were identified in the pull-down RNA that are not predicted targets. We actually identified 1,729 transcripts that were enriched in the pull-down RNA versus control, which is everything on the right side of this figure. In blue, this gene here is not a predicted target, but I like to point it out because this is probably the most significant p-value I've ever seen. It came down with a p-value of 10 to the negative 98. And then all of these other points are colored by their significance, which is the log of the adjusted p-value. We validated some predicted targets and this highly significant gene by qPCR, which showed that qPCR of the total RNA which showed that the expression of the validated genes was downregulated in the total RNA by qPCR from the cells that were treated with mir-486. But then we had to determine which target out of all of these is responsible for mir-486's inhibition of eNOS. And so kind of where do you start? There's 1,729. So we chose to selectively knock out a few chosen targets based on the literature. And the first two that we tried had zero effect on eNOS, completely negative. And then we came across this one called mastermind-like 3 transcription factor, or MAML3, which was significantly enriched in the pull-down RNA. And based on the literature, we decided to try it as well. So for interest's sake, MAML3 is an activator of Notch signaling. Notch signaling is a critical signaling pathway that regulates the critical biological processes, including cell fate, organ formation, tissue repair. And we had also read from another study that MAML3 knockdown inhibited in vitro angiogenesis. So it looked like a good place to start. To see if this could explain our link between mir-486, eNOS inhibition, and inhibition of angiogenesis. And this is this time when we tried it, what we did was we transfected cells with a silencing interfering RNA against MAML3, and this selectively knocks down MAML3 in the endothelial cells. What we saw was that selective MAML3 knockdown significantly inhibited the expression of eNOS, so similarly to mir-486. All right, so in summary, kidney IR injury is associated with a permanent reduction of capillary density, tubular the development of tubulointerstitial fibrosis, and overall systemic endothelial dysfunction. We showed in our rats that early administration of mir-486 significantly protected against ischemic AKI, protected against endothelial injury and inflammation in the kidney. And at 10 weeks, the rats that had been treated with mir-486 at the time of reperfusion displayed no evidence of chronic kidney disease and no global endothelial dysfunction. When we treated rats with mir-486 late after the initiation and extension phases of AKI had developed, we found that mir-486-5p had no impact on long-term kidney and vascular outcomes. However, we also noted that there were no adverse vascular effects despite eNOS inhibition in vivo. In vitro, though, the mir-486 function directly in vitro is a bit of a different story because we showed that mir-486 inhibited eNOS and angiogenesis in cultured endothelial cells. We identified many targets, more than what's actually predicted with the online databases. And of the targets that we've tried, we found that MAML3 inhibition also inhibits eNOS. So we'd like to conclude with the following statements. Based on our data, early intervention with a single dose of mir-486-5p at the time of reperfusion protects against kidney ischemic injury and long-term kidney and systemic complications. Our study suggests that early gene responses are important in programming the transition from AKI to CKD. We also provide data on timing of administration, efficacy, and safety, specifically administering this treatment later in the course of acute kidney injury, after the initiation and extension phases are likely complete, results in no protective benefit long-term against the development of fibrosis. In vitro, we also showed that eNOS inhibition and the anti-angiogenic effect of mir-486 in cultured endothelial cells might be through the inhibition of MAML3. This is a completely new study. This is a completely new pathway to study with potential therapeutic implications for other disorders. And given the involvement of Notch signaling, potentially could be important downstream for diseases like cancers. And I just want to acknowledge everyone who's helped along the way from Dr. Burns' lab and our collaborators, because if it wasn't for help and collaborators, research like this just wouldn't be possible. So thank you.
Fantastic. Thank you, Adrianna, for that overview of what you've been doing for the last few years. I'll open the room for questions. Please unmute yourself and speak up. Greg.
Trying to figure out Zoom. There we go. Thanks for that, Adrianna. I mean, I guess what my obvious question is, what are the next steps? It seems like it would be ready now for phase 1 or phase 2 human studies, especially in like a high-risk cardiac surgery population, like you mentioned. So is that the plans, and what would be the steps to get it there?
Yeah, so that would that's definitely where we'd like to go with something like this, especially now that we have longer-term data on safety. And I should point out that in the non-human primate studies that have been published a couple of years ago, they actually have 17-month data in monkeys in myocardial infarction studies. So there is some safety data on this now in the experimental models. The next step would be to figure out how to deliver it to humans as well, because right now we're delivering it in a liposomal package. And what we do find is that there's actually significant uptake in the liver. Although we don't find any evidence of liver toxicity, that is one of the sites where this is being taken up. So we want to be able to deliver it in something that more selectively binds the kidneys. And there is work going on right now in Dr. Burns' lab. There's a collaboration with Dr. Suresh Gadi, who's a nanoparticle expert, to try to package the mir-486 in FDA-approved nanoparticles that could specifically hone to the injured kidneys and deliver it.
So how are the other human studies that you mentioned in those other microRNAs delivered? Yeah,
so in the monkey study, they're not in the non-human primate study, it was exosomal microRNAs.
And you can't use that in this setting?
The concern with using exosomes clinically, although we've shown in mice that exosomes work, is that they have a bunch of other content. They have a bunch of other RNA content. And so the safety long-term of those would be less clear, and the off-target effects from the other content that are being delivered would be unclear. There can also be a lot of batch-to-batch variability with exosomes. So from a quality assurance perspective, it might be a little bit more difficult to use.
So with this, sorry, one last thing. So would this phase 1 in humans be still years off then? Is that what, to get it into some nanoparticle delivery system? And
so exactly. So the nanoparticles, from what I'm aware of, are FDA approved, which does shorten it, because if you can have preclinical data that shows efficacy, you might be able to go straight then to a human trial. But it needs to take some work to optimize to make sure that you can encapsulate the mir-486 and that it can be released. So that's currently the stage that they're on now. Is the lab has been able to encapsulate mir-486 in different types of nanoparticles, but releasing it into the cell so that it can bind its target is proving to be more challenging.
Thanks.
So.
Awesome. Any other questions from the rest of the audience? We do have about 15 minutes. Oh, conference room, go ahead.
It's Marcel. Adrianna, very nice presentation. I just have one question. How do you explain what is in my mind discrepancy between eNOS inhibition at the same time, no demonstrated endothelial dysfunction?
Yeah, we were puzzled too. So I think there's a couple of answers there. One is that, so at least when you're giving mir-486 at the start of reperfusion, you are preventing the occurrence of kidney injury. You're really minimizing kidney injury. So it's possible that in that setting, there's not enough ischemic injury to induce the upregulation of eNOS. So that, yeah. So potentially there, because of that mechanism, what was that? Yeah. Because we're preventing the injury, maybe we don't need the upregulation of eNOS, is what I meant to say at the beginning. And the half-life of mir-486 is not going to act that long. It acts very quickly on its genes, and then it goes away. So the one-time dose in that critical time point may not be enough to have long-term effects. But you're absolutely right. If you were to give a treatment like this and you were to deliver it in a repeated manner, like in a repeated dosing manner as a long-term treatment, and it has this effect on endothelium, it's possible that you could bring out that adverse endothelial effect later. So it could be a timing and dosing impact. So what we're doing now seems to be safe, but if you were to dose higher or dose more frequently and keep the treatment going on for longer, then you might start to see that adverse impact.
Thank you. And the second question to that would be, wouldn't the muscle vascular bath be better in terms of studies of endothelial dysfunction than the substrate which you used, which I believe was mesenteric?
Yeah. Yeah. We chose, so mesenteric vessels are commonly used in this type of work to look at a global endothelial dysfunction. And that's the expertise that we had at the Kidney Research Center, was in isolating mesenteric vessels, not so much in isolating muscular vessels. And that was one of the other main reasons why we chose to go with mesenteric vessels, was based on the expertise that we had and what we knew we could do.
And we can see it. Oh, thanks, Adrianna. That's fantastic work and a lot of work, obviously. So thank you. So actually, along the lines of what Marcel was asking, like, what about in a septic model because of sepsis? Because that's, you know, that you have global endothelial dysfunction. Yeah. And then if it goes to liver, great. So like, I don't know, is there, you know, if you're dealing with a diffusely septic, like diffuse process, is there a consideration to test it in a septic animal model?
Yeah, we haven't done it yet, but that certainly would be a consideration because we don't know if it would work. And there are septic animal models that you could use, like LPS injection or cecal ligation and puncture, and it could have benefit. We just don't know yet.
Excellent, thanks.
Thank you. Thank you.
Are there any other questions from the conference room? Okay, if not, there is a question in the chat from Dylan. I don't know if you can see it, Adrianna, but I can read it out for the others. So aside from the direct treatment, do you see other ways to leverage the miRNA skills you have gained in the PhD in the clinic? It's a very broad question.
Yeah, so in addition to direct treatment, so like in the clinic, if we're interested in looking at biomarkers for certain specific kidney disease processes, I think some of these skills that I've picked up along the way with the PhD project could go into looking to conducting better biomarker studies that are more clinically relevant than what we've currently seen in the literature. That would be one way, one thing I could see going forward for sure.
Thank you. Are there any other questions from the other people in the audience? I see your PhD supervisor is also in the chat if Kevin wants to make any comments.
Thanks, Swapnil. That was a nice talk, Adrianna. And, you know, I just want to emphasize how much work this was for her to complete these studies. You know, each slide represents
Weeks,
a lot of agony and hours and hours of learning the technique, perfecting the technique. And I would say especially the vascular studies, which were new to our lab, and thanks for the collaboration to help us get those done and help Adrianna do those quite difficult studies. But I'd also emphasize the pull down experiments that she did, and Adrianna came up with this idea from reading the literature. And initially I thought this is going to be a fishing expedition, which it is some degree, but luckily she came up with a target, which is quite interesting. The other thing I'd emphasize is, and Adrianna hasn't mentioned this, but maybe I'll let Adrianna, and this may have been the focus of Dylan's question, is maybe what you're doing in the future and how that might be applied to microRNA research. If you want to comment on that, Adrianna, your fellowship with Dr. Stanford.
So I'm just transitioning now from finishing the PhD to a postdoctoral fellowship in Dr. Stanford's lab, where I'm going to be looking how to work with kidney organoids to model other kidney diseases. That could be a way to bridge like the RNA biology with other kidney disease mechanisms. But I'm specifically going to be studying it in genetic forms of kidney disease to learn how to work with the model. But I'd like to eventually combine the two worlds as my area of expertise. So kidney organoid modeling and RNA biology.
Fantastic. That looks very promising. So congratulations again on your PhD and good luck on your postdoctoral fellowship. If there are no other questions today, we'll wrap up. Thanks again, Adrianna, for giving us a taste of what you have been doing for the last few years. For the people in the audience, there will be an email you will get for the evaluations as well. In addition, if any one of you have any issues with Zoom, please let me know and we'll figure out and fix it before next time. Thank you again.
Thank you, everybody.
See you, Adrianna.
Bye.